Good afternoon, everyone. Angiosperms or flowering plants showcase one of nature's most remarkable processes, the development of gametophytes. Today, we will focus on the female gametophyte, exploring how this intricate structure ensures the reproduction and survival of these plants. Let's begin by briefly understanding their life cycle, which alternates between the diploid sporophyte and haploid gametophyte generations.
Life cycle of angiosperms. As with all land plants, alternation occurs between sporophytes and gametophytes generations. Represented here by the sporophyte plant, the life cycle of an angiosperm alternates between a diploid 2N sporophyte generation and a haploid gametophyte generation. The flower is the mature sporophyte structure that produce gametophytes.
The male part of a flower is called the stamen, which has two parts, the anther, where pollen is made, and the filament, which hold the anther. Female part is called the carpel, which includes the stigma, sticky parts that catch pollen, the style, a tube connecting the stigma to the ovary, and the ovary that holds the ovules. Flowers that have both male and female parts are called bisexual or perfect flowers, like rose and lilies. Flowers with only male or female parts, like those two flowers, are called unisexual, like corn. These parts help flowers reproduce and ensure plant survival. The anther sac is a crucial structure in the reproductive system of flowering plants. It consists of multiple layers that play essential roles in the production of pollen. The epidermis. We have the fibrous layer, which provides structural support. The central part of the anther contains the pollen sacs, where pollen is produced. Inside the pollen sac, we find the pollen mother cells, which undergo meiosis to produce haploid pollen grains. The tapetum, a nutritive tissue, provides essential nutrients to support the development of pollen grains. These pollen grains containing the male gametes are eventually released for fertilization. Pollen grain development. Pollen development occurs within the pollen sacs of the anther. Each sac contains diploid parent cells, microsporocytes, that undergo meiosis to form a tetrad of 4 haploid microspores, and then fall apart, swell, and form more separate 4 pollen grains.
Now, let us talk about ovule and embryo sac development in plants. The process begins in the ovule with a single diploid megasporocyte. This cell undergoes meiosis, dividing into 4 haploid megaspores. Of these, only one survives to continue its development, marking the first step toward the formation of the embryo sac. The surviving megaspore plays a pivotal role as its nucleus undergoes 3 rounds of mitotic division, producing 8 haploid nuclei within a single large cell. These nuclei then migrate and organize into distinct groups. 4 move to each pole, while 2 from each group merge at the center to become polar nuclei, later crucial for endosperm formation. The result is the mature embryo sac, which consists of 7 cells and 8 nuclei. At the micropylar end, you find 3 nuclei, 1 egg cell flanked by 2 synergids. In the center are the 2 polar nuclei, and at the chalaza and reside 3 antipolar cells. Together, These structures create a highly specialized environment, primed for fertilization and subsequent development. The ovule is a structure in flowering plants that develops into a seed after fertilization. It consists of the funicle, stalk attaching to the ovary wall, micropyle, a small opening for the pollen tube, integuments, protective layers that form the seed coat, and nucellus tissue nourishing the embryo sac. Inside the ovule is the embryo sac, which contains 3 antipodal cells at the chalaza end with unclear function, 2 polar nuclei in the center, which form the endosperm after fertilization, 1 egg cell, the female gamete that fuses with sperm to form the zygote, and 2 synergid cells near the micropyle guiding the pollen tube. Those called the mega gametophyte.
Now let us have a small summary. A single diploid megasporocyte exists in the ovule and divides by meiosis to produce 4 hybrid megaspores, only one of which survives. The surviving megaspore divide by mitosis to produce 7 haploid cells. The 7 cell structure make up the female gametophyte called the megagametophyte. Single diploid microsporocyte exists in the anther and divide by meiosis to produce 4 haploid microspores. By producing 2 different types of spores, the microspore and megaspore, angiosperm, and all other seed plants are considered heterospore. Each microspore undergoes a mitosis division. Haploid microspore after 2 times of mitosis consists 2 sperm cells and 1 vegetative cell. The pollen grain germinates and a pollen tube rolls down the stem until it meets the female gametophyte. 2 sperm cells from the pollen grain travel through the pollen tube and enter the tube and the female gametophyte. One fertilizes the egg, forming a diploid zygote. The other fertilizes 2 pollen nuclei, forming a triploid cell. The fertilization of both egg cells and the central cell called double fertilization, a hallmark of the life cycle of angiosperm.
Seed and sporophyte development. The zygote develops into the embryo. The triploid cell becomes the endosperm. Once the seed germinates, the sporophyte matures, and the life cycle begins again.